In this talk, we're going to discuss how molecules move across the cell membrane. And really what we're answering in this talk is, how is the cell membrane selective? So we want to answer this question because we know that the cell membrane is selective, or it's semi-permeable, but how does it do that? So in this talk, we'll discuss how simple diffusion helps the membrane to be selective and how facilitated diffusion helps the membrane to be selective. These are two types of diffusion that we need to know. So remember that the cell membrane is selectively permeable. It's selectively permeable. It selects what goes in and out. Another way to say that is semi-permeable. And one of the main concepts that you need to kind of remember is, remember how polar dissolves polar and non-polar dissolves non-polar. Another way to remember that is that polar, or I guess non-polar, repels polar. Okay? It repels. And what does that mean? It means that it pushes something away. To repel means to push away. So the other thing we need to remember is that the inside of this membrane here, that the interior of the membrane, right, here's the interior, it's a non-polar interior. It's a non-polar interior. So there's no water in here. And it doesn't really like polar substances in there. But we'll talk a little bit about that in a minute. But it really doesn't mind if non-polar substances go through it. Now, remember that the phosphate heads, this part here, these parts here, and these parts here, these are polar. And so they like to be in the water, but they're a little bit afraid of non-polar. So let's have a look at some things that can go through and some things that can't go through. So some things that can go through are things like gases. And the gases that are often moving through our cell membranes are things like oxygen and carbon dioxide. Now remember, these are both non-polar molecules, but they're also very, very small, right? So the size matters and the polarity matters. Okay, so it's determined by polarity and size of the molecule. Okay, so we need to consider both of those things. So they're small and they're non-polar, and so for that reason, they're able to go through. What they do is they're actually able to kind of push these polar heads away and go through. Benzene, this is an example of a very non-polar. It's large, but it's very non-polar. Okay, and so again, it's going to repel these phosphates, these phospholipids, the phosphate heads, away. And for that reason, as it pushes those phosphate heads away, it's able to move through that hydrophobic interior, right? Because it's non-polar. The inside of the cell membrane is also non-polar, so it's able to go through. Now, the other thing that's able to go through is small polar molecules. So these are small, obviously, and they are polar. But they're able to go through. So they're able to kind of squeeze through. Not sure how easy it is, but they're able to sneak through somehow. So the main one that you need to know is water. Water is able to move through the cell membrane all by itself. The other thing is, a type of alcohol called ethanol. Ethanol can also get through. That's maybe about as big as it gets. I'm not sure how much larger of a polar molecule can get through the cell membrane, but some small polar molecules can get through. Now, when it comes to large polar molecules, remember these are big in size, so they're large and they are polar. They do not get through. They get repelled. They get repelled by the cell membrane. Same thing with anything that carries a charge. So here we've got charged ions. Some amino acids might carry a charge, right? And if those amino acids carry a charge, they get repelled. They do not get through. They get repelled by the cell membrane because the hydrophobic part, the hydrophobic interior, repels them. So even though they're able to interact with the polar heads, they like the polar heads of the phospholipids. They're afraid of the lipid tails. They do not like the hydrophobic interior. So all of these molecules here all of these ones here, they are repelled. They are repelled by the non-polar interior. Okay, so they don't like that they can't get through that hydrophobic interior. They're repelled by that. Okay, so gases are able to go through. Gases are able to go through. Hydrophobic molecules are able to go through. Small polar molecules are able to go through. Large polar molecules are able to go through. But large polar molecules and charged molecules are not able to go through. Let's give these a little highlighting. Gases, yes. Gases, yes. Hydrophobic molecules, yes. Small polar molecules, yes. But then large polar molecules, no. And charged molecules, no. They get repelled. Okay, so there are different types of transport across the cell membrane, and we're going to talk about the types of diffusion today. So we're going to talk about simple diffusion and facilitated diffusion, and these are examples of passive transport. The other type of passive transport is osmosis, but we're going to leave this for another talk. We are also going to talk about active transport. We're going to talk about active transport, and we'll talk about bulk transport. Bulk transport is a type of active transport, but it's specific because it takes in so many molecules. And there's two types that we're going to talk about, endocytosis and exocytosis. For active transport, we're going to give the example of the sodium-potassium pump, and I think we're also going to talk about the hydrogen ion pump. So the thing to know with diffusion is that molecules are moving through the cell membrane from high concentration to low concentration. So sometimes they move across without a protein, and sometimes they move across with a protein. If it's without a protein, that's called diffusion. If it's with a protein, it's called facilitated diffusion. Really, this would be called simple diffusion. And these are both types of passive transport. So what is diffusion? Diffusion is just the tendency of a substance to spread out through an area, and the molecules, they want to spread out equally in an area. They don't want to crowd. They want to be spaced out equally in a certain area. So over time, if you start with molecules that are together like this, over time what's going to happen is that these molecules are going to start to spread out in the solution, okay? And eventually what you'll get an equal concentration, an equal spacing of those molecules, so an equal concentration all around. And so what we say is that molecules, they move from an area of high concentration, so over here, this is the high concentration side, to an area of lower concentration, and this is the low concentration side. So the tendency of the movement of particles is to move in this direction here, right? Molecules are going to move in this direction from left to right. And just as a reminder of what concentration is, sometimes we forget what that is. Concentration is usually defined as a mass over a volume, right? So usually we might talk about concentration in terms of like grams per liter. Or we might talk about it in another way to say about it, which you'll learn in chemistry, is moles per liter. Okay, sometimes we can also have a volume over a volume. Remember, you can have a solution that's two gases mixed together or a mixture of two liquids. And so in that case, you'd usually have like some sort of a percentage volume. Okay, so now that we understand that diffusion is molecules moving from high to low concentration, let's talk a little bit about simple diffusion. So this is a type of passive transport, as we mentioned, and it does not require energy. It does not require any protein to help things move across, and particles move along their concentration gradient. And concentration gradient is one of those key terms that you need to understand. And what that is a concentration gradient is a difference in concentration. Okay, so it's a difference in concentration. So here's our membrane here, right? You can see there's our membrane, and it's divided into, you can see that there's gaps, there's spaces for molecules to move through. And you can see over here we've got these molecules, and there's more molecules in the same volume compared to this side over here, which has none. It's just pure water. So this side over here has all these solute molecules, and over here it's just water, the solvent. And what's going to happen is that those molecules are going to move from where it's high concentration to low concentration. They're going to move across that cell membrane until gradually there is an equal concentration on both sides. And once the concentration is equal on both sides, then the molecules they appear to no longer be moving. And so we call that equilibrium, because they're equal concentrations on both sides, equilibrium. But what's important to understand is that it's not done. It doesn't mean that there's no more movement. Okay, molecules are going to be moving in both directions now. They're going to be moving equal and opposite in both directions. So they might be moving this way and this way in that equilibrium. And so we call this a dynamic equilibrium. Okay, we call this a dynamic equilibrium. Dynamic means that it's still moving, right? So again, like you can see here, dynamic equilibrium, what is this? This is a key term that you need to know, just like concentration gradient. It's when the concentration is equal on either side of a membrane, but the molecules are still moving in both directions. They're moving equally in opposite directions. Maybe we should say equal movement in opposite directions. Now, one thing that we should maybe just describe here is this idea of moving along a concentration gradient and against a concentration gradient. So from high to low concentration, okay, so when you're going from high concentration to low concentration. That's called moving along the gradient. Okay, and then against the gradient is the opposite. Against the gradient is from low concentration, I'm just going to put CONC, to high concentration, CONC. Okay, so this is kind of, you need to be able to use those phrases there. Okay, so substances are going to move across the membrane from high solute concentration to low solute concentration. And again, these are things like nonpolar molecules, small polar molecules, gases like oxygen and carbon dioxide. And they are moving along their concentration gradient, right? They are moving along the concentration gradient. Okay, and they go directly across the cell membrane. They don't need a protein. They're just moving directly across, and there's no energy. No energy being used. Okay, now facilitated diffusion is a type of passive transport as well. Again, it does not require energy. It does require a protein to move. Okay, so there's two types of proteins that we need to know: protein channel and a carrier protein. Okay, this is a protein channel, or a channel protein is kind of like, it's like a tunnel. Okay, so here's the protein channel, and you can see that it's kind of like a straw or a tunnel. It's moving from one side to the other. Whereas a carrier protein, it changes shape. Okay, it changes shape. So this is going to be open to this side sometimes, and it'll be closed on this side. And then it will open up, and then when this molecule moves in, it changes shape so that it's constantly moving back and forth. And what it does, it just helps larger molecules, especially things like large polar molecules or ions, things that are really strongly repelled by that hydrophobic interior. Remember, these things are repelled by the hydrophobic interior, so they need help to go through the cell membrane, and they need a protein to do that. And again, they're moving along the concentration gradient from high concentration to low concentration. Okay, so these are things like ions. These are things like large polar molecules. They require that. So specifically, channel proteins, they typically help charged particles. So things with a positive or a negative charge might need to go through a channel protein. And so, yeah, it's like a tunnel that kind of connects the extracellular environment to the cytoplasm. It's helping those charged ions. It's helping those ions move from high concentration to low concentration. So things like sodium ions or potassium ions or chlorine ions or calcium ions, these are all things that need to get across the cell membrane. They would do that by a channel protein. And then here's the example of carrier proteins, and you can see how they're changing shape here. This shape is open to the extracellular environment, and this shape here is open to the cytoplasm. And so these carrier proteins, they're going to be helping things like amino acids, sugars, small proteins, those sorts of things. These things are typically our large and polar molecules. Okay, so how does this work? Particles will bind, they will stick into the carrier protein on one side of the cell. That's going to cause the protein to change its shape. And then what's going to happen is the particle will be released on the other side of the cell. And then the process just repeats. It goes back to here, and we start that process again. And the cycle continues.